We've all been there, right? You're faced with some complex new topic, maybe sifting through endless articles,
Mm-hmm.
just trying to figure out what really matters.
Mm-hmm. It's easy to get completely lost.
Exactly. And that's kind of the challenge. But it's also our mission here at the Deep Dive,
Mm.
to cut through all that noise
Yeah.
and get you straight to those surprising, impactful insights.
And that's absolutely the aim. We want to help you get informed and quickly by uncovering those aha moments that are often buried pretty deep in the data.
So today we're taking a deep dive into a really fascinating new scientific publication. It's the Pub of the Month, June 2025, and specifically an article called Unfolded von Willebrand Factor Binds Protein S and Reduces Anticoagulant Activity.
Okay, yeah, that sounds technical.
It does sound technical, but don't let the terms put you off. This isn't just some obscure biology experiment. This is about understanding a critical sort of newfound mechanism that really sheds light on why some severe infections can drastically increase your risk of dangerous blood clots. So this has direct implications for, real-world health.
Right. So to set the stage, maybe let's introduce the two key proteins here, the main players in this discovery.
Good idea.
First up, we have protein S, or just PS. You can think of it as one of the unsung heroes in your blood. It's a critical anticoagulant protein.
Anticoagulant meaning it stops clots, prevents them.
Exactly. Its job is basically to help prevent excessive clotting, keeping your blood flowing smoothly. Now, PS circulates in, well, mainly two forms. About 40% is free PS, that's the active anticoagulant form.
Okay, the working form.
The working form, yeah.
Right.
And the rest is bound up with another protein called C4BP, which largely blocks its activity. So it's mostly inactive when bound.
Okay, got it. Free PS is the hero. And the other player.
And then there's our other key player, von Willebrand factor, or VWF. Now, this protein is usually known as a major heavyweight when it comes to forming clots. It's crucial for stopping bleeding when you get injured.
Right, VWF is the one that rushes in to plug the leak, so to speak.
Precisely. But here's where it gets interesting. This study reveals a completely new side to VWF's story.
So VWF, usually the body's like super cup for stopping bleeding, is now maybe acting like a bit of a rogue agent in some situations, disarming the anti-clotting system. That does sound like a plot twist.
It really is, and it potentially explains a long-standing mystery. You see, for a long time, doctors have noticed something perplexing.
Which is?
In many severe viral infections, think HIV, dengue, varicella, and more recently, SARS-CoV-2, which causes COVID-19 patients, often develop this thing called acquired free PS deficiency.
Acquired meaning they weren't born with it. It happens because of the infection.
Exactly. And this deficiency, this lack of active protein S, is directly linked to an increased risk of thrombosis, those dangerous blood clots. But the exact cause why the free PS levels drop has been, well, a bit of a black box.
Ah, okay. So the virus has somehow caused this PS deficiency, increasing clot risk, but we didn't know how.
Precisely. Until now, perhaps.
And this is where the current study comes in, right? This is the core discovery.
This is it. And it's, frankly, pretty astounding. What they found is that VWF, specifically when it unfolds under certain mechanical shear forces in our blood vessels.
Shear forces, like the friction or turbulence of blood flow.
Exactly, like turbulent flow. When
Right.
VWF unfolds under that stress, it directly binds to that active, free protein S.
Wow, okay. So VWF changes shape and grabs onto the free PS.
Yeah.
A direct interaction we just didn't know about.
We didn't. And so what here is truly significant? This binding effectively sequesters that free PS. It pulls it out of circulation, makes it less available.
Right.
And crucially, this reduces its anticoagulant activity. So the paper proposes this newly discovered interaction as a key contributor, maybe the key contributor to that increased risk of blood clots we see in many virus-associated conditions.
Okay, that connects the dots. The virus causes inflammation, maybe more turbulent flow, VWF unfolds, grabs PS, and boom, less anti-clotting action.
That's the hypothesis in a nutshell.
So get ready, everyone. We're going to unpack the elegant experiments that revealed this interaction, see how it actually plays out in real patient samples, including those from COVID-19 patients, and really understand why this finding could fundamentally change how we think about blood clotting and inflammation.
Yeah, I think you'll find some genuinely insightful takeaways here.
This sounds groundbreaking, definitely. But I mean, how on earth did they even start looking for this?
Yeah.
How do they find proteins that interact with VWF, especially when it's under these shear forces? What was the first step?
Well, they used a pretty clever biochemical approach, actually. They took VWF, the protein, and attached it to tiny little beads.
Okay.
Then they took plasma samples, the liquid part of blood, and exposed the plasma to these VWF-coated beads. But here's the key part. They needed to introduce that shear force.
Right. To mimic the turbulent blood flow. How did they do that in a tube?
Surprisingly simply, they just use vortex mixing, spinning the samples rapidly for about 30 minutes.
Just vortexing.
Yeah, simple but effective for inducing VWF to unfold. And what they discovered was, well, striking. The vortexing dramatically increased the quantity of protein S that bound onto the VWF beads.
How much of an increase?
In some samples, it was over a 1,000,000-fold increase compared to non-vortex samples.
1,000,000. Okay, that's not subtle.
Not at all. It strongly suggested that this shear-induced unfolding of VWF exposes some kind of binding site that protein S can grab onto. And crucially, they found it was primarily the free protein S that bound.
Not the PS already tied up with C4BP.
Exactly. Suggesting that PS already bound to its usual partner, C4BP, didn't really participate in this new interaction with VWF.
Okay, that's really strong lab evidence for the interaction happening when VWF unfolds. But like you said, the body isn't a vortex mixer. So the next big question must have been, does this happen under real blood flow conditions?
Absolutely. That was the critical next step.
Yeah.
They needed to move beyond the test tube.
And how did they test that? Could they actually see it happening?
They could, using something called microfluidic systems. These are basically tiny devices like labs on a chip that have microscopic channels designed to mimic blood vessels.
Very cool.
Yeah, they allow researchers to control flow conditions really precisely and observe things happening in real time. So they perfused VWF and fluorescently labeled PS protein S that glows into these devices.
So they could watch where the PS went.
Exactly. And what they observed was that PS stably bound to VWF as the VWF was unfolding under turbulent flow conditions within those microchannels. They could literally see them sticking together where the flow was disturbed.
Wow. And you mentioned calcium earlier. What role did that play?
Yes, that was another fascinating piece. They found that this PS binding to the unfolded VWF was significantly higher when there was no calcium present.
Higher without calcium. That seems counterintuitive somehow.
It does a bit, but when they added calcium back, the binding decreased. The thinking is that calcium might help stabilize VWF in its normal folded state, sort of preventing it from opening up and exposing that PS binding site. Or maybe calcium affects PS itself in some way.
Interesting. So low calcium might actually make this VWF-PS interaction more
I
likely.
think that's what the data suggests, yeah.
Okay, so turbulent flow, check. Low calcium, check. But what about just normal blood flow?
Yeah.
Like in most of our arteries where it's smooth or laminar. Does this complex form there too?
Good question. Under those normal, smooth, laminar arterial conditions, they found that the flow alone did not induce the complex formation. VWF didn't seem to unfold enough just from that smooth flow.
Okay, so turbulence is really the trigger.
It seems to be the trigger for formation, yes. However, they did another experiment. What if the complex was already formed? Say it formed in an area of turbulence.
And then flow downstream to a calmer area.
Exactly. So they preformed the complex unfolded VWF bound to PS and then introduced that into whole blood under normal arterial flow conditions where a clot was forming on platelets.
And what happened?
The VWF and PS stayed together. They colocalized on the growing platelet thrombi, the clot.
So once formed in turbulence, the complex is stable enough to travel and stick to a developing clot even under normal flow.
Precisely. It indicates that once this complex forms, perhaps in an area of inflammation or vessel damage causing turbulence, it's stable and can then circulate and contribute to clotting processes elsewhere.
Okay, so we've established that this binding happens, it's triggered by turbulence, maybe helped by low calcium, and it's stable once formed. But now for the million-dollar question, what's the impact? Does this binding of VWF to protein S actually cripple PS's ability to prevent clots? I mean, that's its whole job, right?
It absolutely does. And this, I think, is where it really hits home in terms of biological significance. They directly measured the anticoagulant activity of PS.
How do you measure that?
They used plasma thrombin generation assays. These basically measure your blood's potential to form clots, how much thrombin, a key clotting enzyme, can be generated.
Okay.
Normally, protein S works with another protein called APC to dampen down thrombin generation. So they took pooled normal plasma and saw that adding this activator, APC, reduced the peak thrombin generated by about 72%. That's PS doing its job well.
Right, putting the brakes on clotting.
Exactly. But then they took the same plasma and vortexed it, inducing the VWF unfolding and binding to PS.
Ah, mimicking the shear stress effect.
Right. And then they measured thrombin generation. That reduction by APC dropped significantly, down to only 56%.
So less braking power. The anticoagulant effect was weaker after vortexing.
Precisely. The ability of PS to act as an anticoagulant was measurably reduced. And crucially, they showed this effect was entirely dependent on both PS and VWF being present.
How do they show that?
They used plasma from patients with a condition called type 3 von Willebrand disease. These patients completely lack VWF.
Okay, no VWF in their blood.
Right. And when they vortexed that plasma, the protein S activity wasn't altered at all. It still worked just as well as before.
Wow. Okay, that nails it down. It's the unfolded VWF binding the PS that reduces its activity.
That's the conclusion, yes. The unfolded VWF binds the free PS, reducing its concentration and directly decreasing its crucial anticoagulant function.
This feels like a really profound shift in understanding VWF's role. So it's not just some theoretical interaction in a lab dish. It has real measurable consequences for clotting potential.
It certainly seems so.
Did they manage to see evidence of this mechanism playing out in actual patients? You mentioned COVID-19 earlier, where this acquired free PS deficiency is known to happen. Did they look there?
Yes, and this is obviously a critical piece for connecting the lab findings to human disease. They analyzed plasma samples from a group of healthy controls and compared them to patients hospitalized with severe COVID-19.
A condition definitely known for free PS deficiency and, sadly, a high risk of blood clots.
Exactly, and what they found was quite compelling, just as expected, while the total amount of protein S was pretty much unchanged between the groups.
Okay.
The levels of free active PS were significantly decreased in the COVID-19 patients compared to the healthy controls.
Right, the known deficiency. But could that be explained by the usual suspect, the C4BP protein maybe going up and binding more PS?
That's what they checked next. But no, they found that this free PS deficiency could not be explained by changes in C4BP levels. The usual suspect wasn't the culprit here.
Interesting. So if it wasn't C4BP tying up the free PS, what did they find in the COVID-19 patients that could explain it based on their new hypothesis?
Well, just as other studies have reported, they found that plasma VWF levels were significantly elevated in the COVID-19 patients.
Higher VWF, which makes sense with inflammation and endothelial activation in COVID.
Right. And here's the really key finding. VWF levels negatively correlated with the free PS concentration.
Meaning, as VWF went up, free PS went down.
Precisely. The more VWF a patient had, generally, the less free PS they had available.
Wow, that's a strong link in patients.
It is. And furthermore, they looked at the functional consequence, that thrombin generation potential we talked about. Even though many patients were already on heparin, a common blood thinner, their plasma still showed significantly elevated thrombin generation potential, especially when measured in a way that highlights the protein S pathway.
So even with anticoagulants, their underlying clotting potential related to PS dysfunction was high.
Yes. And this reduced PS anticoagulant activity strongly correlated with those lower free PS levels. So the data from the COVID-19 patients lined up perfectly with the mechanism they'd uncovered in the lab.
Increased VWF, likely unfolded due to the inflammatory state, binds the free PS, reduces its availability, hinders its function, leading to higher clot risk.
That's the story the data tells.
That's incredibly powerful, tying it all together from the beads and microfluidics right through to clinical observations in severe disease.
Let's maybe talk a bit more about the nitty-gritty of the binding itself. Did they figure out where exactly on these big proteins this interaction is happening? They did delve into that, yes.
Is it like interfering with other known binding sites on PS or VWF?
Good question. They managed to pinpoint the region on protein S involved. They showed it was a specific site, and importantly, it seems distinct from where other key partners, like that activated protein C, APC we mentioned, usually attach.
So VWF binding doesn't directly block APC binding, perhaps?
It seems not directly, although by reducing free PS, it indirectly affects the whole pathway. They also found that another protein, TFPI, actually competed with VWF for binding to PS.
Okay, so TFPI and VWF might be fighting for the same spot or nearby spots on PS.
Something like that. It helps narrow down the general area on PS involved in this new interaction. And similarly, on the VWF side, this new interaction site for PS appears distinct from the well-known site where platelets bind.
Ah, so binding PS doesn't necessarily stop VWF from binding platelets and vice versa.
It seems that way. They showed that treatments designed to expose the platelet binding site on VWF didn't affect PS binding. And conversely, the shear forces they used to expose the PS binding site didn't seem to stably expose the platelet binding site.
Okay, so it really emphasizes that this is a specific, distinct interaction between VWF and PS, separate from their other known jobs.
Exactly. It highlights the specificity of this newfound VWF-PS interaction.
Right. Let's try and recap the core mechanism then. We've explored how this specific newly discovered interaction between von Willebrand factor, VWF, and protein S fundamentally changes the picture of blood clotting.
Mm.
The absolute key takeaway seems to be that when VWF unfolds, which happens under mechanical shear forces, like the turbulent blood flow you might get in areas of inflammation or vessel damage.
Right, when it changes shape due to stress.
It suddenly reveals a previously hidden binding site specifically for protein S. It's almost like VWF was, I don't know, hiding this ability until the conditions were right, that unfolded shape being the trigger.
Yeah, that's a good way to put it. And this newly formed VWF-PS complex has two really critical consequences. First, it effectively grabs that vital anticoagulant, the free protein S, pulling it out of active circulation. That leads directly to the deficiency we see.
So it lowers the amount of the good guy.
It does. And second, perhaps even more crucially, it directly reduces the ability of the PS that is bound to actually do its job to prevent clots.
So it reduces the quantity and the quality, so to speak.
You could say that, yes. And this quite elegant molecular dance really helps explain that puzzling acquired free PS deficiency that's seen in severe viral infections. They mentioned HIV, dengue, varicella, and of course, as the study showed explicitly, in COVID-19 patients.
Right. Just to reiterate, those COVID patients had the elevated VWF, which inversely correlated with their reduced free PS levels, and functionally, they showed that increased thrombin generation potential, indicating weakened anticoagulation.
All consistent with this mechanism.
And this really hammers home the novelty here. This isn't just some minor adjustment to the textbook diagrams. The study proposes this represents a heretofore unrecognized procoagulant activity of VWF.
Exactly. We always think of VWF as procoagulant because it builds clots by binding platelets and collagen. But this is different.
Yeah, this is VWF acting in a procoagulant way by actively disarming one of the body's natural clot busters, protein S. And the authors state, to their knowledge, this is the first description of anticoagulant regulation by VWF.
A completely new function attributed to this well-studied protein. It's quite remarkable.
It really is. And, this discovery doesn't just answer one question about viral infections and clots. It feels like it opens up a whole bunch of new ones, doesn't it?
Oh, absolutely. It prompts a lot of further thought. For instance, consider this. Other factors are known to influence VWF unfolding, or PS levels. Things like hypocalcemia, low calcium levels.
Which you mentioned earlier, and which was also reported in some COVID-19 patients, right?
Exactly. And also things like hypertension, high blood pressure, which involves shear stress changes. These factors are known to influence VWF and PS. So you have to wonder, what might be the broader implications of this VWF-PS interaction?
Beyond just viral infections.
Yeah, in various other inflammatory conditions where VWF might be elevated or unfolded, or maybe even in normal hemostasis, the process of stopping bleeding at sites of vascular injury where there's always some turbulence.
Could this interaction play a role even in normal clot formation and limitation?
It's certainly possible. This dynamic interplay they've uncovered between a procoagulant factor, VWF, and an anticoagulant protein, PS, it could potentially offer completely new targets for therapies down the line.
Or just new ways to understand really complex disease states where inflammation and clotting go hand in hand.
For sure. And it really makes you wonder, doesn't it? What else might VWF be doing in our bodies, perhaps depending on its conformation or the stresses it's under, that we just haven't discovered yet?
A fascinating thought to end on. It seems like even well-known proteins can still hold some surprising secrets.